sequencing platform that we are very experienced, and we are very excited to host that for a number of years. It's the Illumina Sequencing platform. And that is a very high throughput sequencing platform. That what we use that is really to generate a whole genome sequence of viruses and bacteria. So for example, we have generated thousands of whole genomes of HIV, also of hepatitis C, but we're also generating a lot of whole genomes from TB and drug resistance strains of tuberculosis, but also other bacteria. The Illumina platform that we have in our library, it's a MySeq, so that's very optimal to generate genomes of lower organisms, this means viruses and bacteria. But as part of a national collaboration program that we call the Diplomics, we also have access to the biggest machines of Illumina. This means NextSeq and HiSeq. And it's quite exciting because now what we have been producing is a lot of like transcriptomic data and exomes data with Illumina sequencing machine, but we are also coming in a collaboration to produce like a lot of whole genomes of mammalian species that are present in the African continent. So we are just validating some methods to sequence, for example, whole genomes of cheetahs and rhinos, as part with a strong collaboration that we are developing with the Agriculture and Environmental School at University of KwaZulu-Natal. So sequencing whole genomes of viruses are very important to answer very important scientific questions. The first one, is this virus, where is it coming from? What's the transmission pathway of them across the population? And we just got quite a high impactful paper that was published in the Lancet HIV that we use that to understand the cycle of transmission of young women. We also use these machines to produce whole genome of drug resistance organisms. For example, we use that on HIV drug resistance. So that's used to identify the best treatment for individuals, and also from TB drug resistance. And that's very important because the current methods to identify drug resistance on TB is take weeks and weeks to identify, and this machine can produce data very quick. In a couple of days you can get the response of which drugs will work on the patient, and then tailor-made that for what we call precision medicine, best treatment with the most appropriate medicine for the patients.